Welcome to The Explainer. Today, we're diving into the absolute ultimate biological manufacturing process, translation. Have you ever wondered how the genetic code locked away in your DNA physically turns into the proteins that build, well, you? You're in exactly the right place. We aren't just going to look at chemical reactions here. We're taking a tour of a high-speed, highly coordinated cellular factory. It is literally a masterpiece of biological engineering. Let's break it down.
So here's our factory tour itinerary for today. We'll start with the blueprint, move into pre-production, check out the machinery, and then walk through the three main assembly phases: initiation, elongation, and finally, termination.
Part 1. The Protein Factory Blueprint. All right, let's kick things off by figuring out exactly what this biological factory is building in the first place and what instructions it's using to pull it off.
You know, when we hear the word translation, we usually think about converting English to Spanish, right? But in biology, translation is the super energy-intensive process of linking amino acids together to form a final polypeptide product, which is a protein. It's a process of polymerization. And the sequence of these amino acids? It is absolutely not random. It's strictly dictated by the sequence of bases in the messenger RNA, or mRNA. Think of mRNA as the master instructions that have been safely walked out of the nucleus and straight onto the factory floor.
To really wrap our heads around this, we're going to map these microscopic biochemical terms directly onto a high-tech manufacturing plant. So the mRNA, that's our factory blueprint. The ribosome is the heavy machinery doing the actual assembly. The amino acids are our raw parts. The transfer RNA, or tRNA, act as our highly specific delivery trucks. And the final polypeptide chain, that's our finished product rolling right off the line. If you keep this analogy in mind, the complex molecular biology we're diving into is going to make total sense.
Moving on to Part 2, pre-production, or charging the tRNA. Look, before any assembly line can actually start running, we need our delivery trucks loaded up with the exact right materials, right?
And the tRNA structure itself is just fascinating. It's literally an adapter molecule. In a 2D drawing, it looks kind of like a clover leaf, though in actual 3D space, it's a compact molecule shaped more like an upside-down L. But its function is basically twofold. On one end, you've got the anti-codon loop, which has bases perfectly complementary to the code on the mRNA blueprint. Then, at the completely opposite end, there's an amino acid acceptor end, so it reads the code on one side and holds the physical raw material on the other. It is the ultimate adapter.
But, those delivery trucks aren't just going to load themselves. This pre-production phase involves a specific process called charging, or, if we want to get technical, aminoacylation of tRNA. Because forging peptide bonds later on is going to take a ton of energy, the amino acids are first activated using ATP. That energy is used to physically link a specific amino acid to its specific cognate tRNA. Bam! Our delivery trucks are now fully loaded, charged with energy, and ready to drop their parts off at the assembly line.
All right, Part 3, the assembly line machinery. Let's step onto the factory floor itself and check out the core machinery powering this whole operation. The ribosome is the cellular factory for synthesizing proteins. It's a massive complex built out of structural RNAs and about 80 different proteins. Now, when it's inactive, it just sits there as two separate pieces, a large subunit and a small subunit, just waiting for a translational unit of mRNA. And this mRNA unit is very particular. It's flanked by a start codon, which is always AUG, and a stop codon. But it also has these untranslated regions, or UTRs, at both the 5' end before the start codon and the 3' end after the stop codon. These extra sequences don't get translated into protein, but they are absolutely crucial for the factory to run efficiently.
Here is the really wild part. The ribosome isn't just structural metal holding things in place, it's an active machine. To link amino acids, a peptide bond has to be formed. Now, if two charged tRNAs are brought close together, forming that bond is energetically favored, but it still needs a spark. The ribosome is that spark. In bacteria, a specific structural RNA right inside the ribosome, the 23S RNA, actually acts as a catalyst. We call this a ribozyme, literally an RNA enzyme. It actively speeds up the rate of peptide bond formation so our factory can crank out products at lightning speed.
Part 4, initiation, or starting the line. Okay, raw materials are charged, machinery is powered up, it is time to flip the switch.
Initiation officially kicks off when the small subunit of the ribosome bumps into our mRNA blueprint. It scans along the line until it locks right onto that specific start signal we mentioned earlier, the AUG start codon. And this start signal is recognized by a very unique delivery truck called the initiator tRNA. Once that initiator tRNA binds to the AUG, the large subunit clamps down hard on top, and boom, the factory is officially open for business.
Which brings us to part 5, elongation. We are now in the main production phase, where the biological product is rapidly assembled.
During this elongation phase, the large subunit of the ribosome provides specific parking spots for the tRNA delivery trucks. It perfectly aligns two tRNAs right next to each other, so the amino acids they're carrying are sitting just close enough to bond. Then, that 23S RNA catalyst we just talked about goes right to work, instantly forging the peptide bond between them.
The mechanics here are amazingly rhythmic. The tRNA delivery trucks pull in one after another, matching their complementary anticodons to the specific codons on the mRNA blueprint. Once an amino acid is added to the growing chain, the ribosome physically ratchets down the mRNA to the next codon. The empty truck drives off, a new loaded truck pulls in, and the amino acids are added one by one, perfectly translated from the exact sequence dictated by your DNA. Finally, part 6, termination. Just like a real-world factory, every assembly line has to have an endpoint, right? Otherwise, the machinery would literally just run off the tracks.
So, the factory keeps ratcheting down the blueprint until it hits a stop codon. And here's a totally fascinating detail. There are absolutely no tRNA delivery trucks that carry a matching anticodon for a stop codon. None. So instead of a truck pulling into the parking spot, a specific protein called a release factor binds to the stop codon. This basically jams the gears in a perfectly controlled way, terminating the translation process. The machinery disassembles, and the finished, fully completed polypeptide chain is released to go do its job in the cell. It really leaves us with a staggering reality to ponder. We've just mapped out a fully automated, incredibly precise manufacturing plant, complete with blueprints, adapters, heavy machinery, and enzymatic catalysts. And this flawless, high-speed molecular factory is operating millions of times per second inside you at this very moment, just to keep you alive. It's constantly translating the code of your DNA into the reality of your biological self. So, how does knowing all of that change your perspective on the sheer machinery of life? Pretty mind-blowing, right? Thanks for hanging out with me for this explainer, and definitely keep exploring the incredible mechanics of life.